Serventi, thank you. Oh, we'll start recording this. Thank you for being here with us. So a little bit of background on Dr. Serventi. So he is originally from Parma, Italy, where he started his university studies in food science and technology. Then he visited OSU for a 6-month experience. He liked it so much that he stayed for his whole PhD in the Votivot's lab, working on saponins from legumes as a tool to reduce cholesterol absorption and improve food texture. He was also a part of the Coccobello team that won the product development competition in 2010 at IFT, and he was the president of the Food Science Club that same year. He had industry experience in R&D at an Italian gluten-free company in Trieste, supporting the launch of their first American factory in New Jersey. The passion for research brought him to Denmark for a postdoc at Aarhus University, learning about sensory, bakery enzymes, and sustainable food products, cassava bread for South America. And since 2015, he is involved with teaching and research in New Zealand, specializing in Liluva, upcycling the wastewater of legume processing into texturizers, sources of bioactives, and packaging. So thank you for being here with us. And if you want to get started, you should be able to share your screen, Dr. Serventi, when you're ready.
All right. Thank you very much, Katie, for the wonderful introduction. Can you all see my screen?
Yes, we see. We can see it, but it's not in presentation mode.
Okay.
We're seeing your presentation mode now. Yeah.
Oh, perfect. Yeah, just a little bit slow. Thank you very much.
We're looking your presentation. We don't see the.
Yeah, so we can see the next slide too.
We see the speaker's view.
Yeah, the speaker view.
Oh, I see. See if I can change this.
You can go to display settings and swap.
There you go.
Ah.
Perfect.
Now we're cooking with gas.
Perfect. All right, so I'll get started. It's a pleasure to be here. It's been a few years since I was at OSU, and I always kept in touch with colleagues and friends over there. And today I wanted to share with you my passion for this topic. And the presentation would be particularly about legume water, but in our research group, we also work on other byproducts like okara from soy milk or defatted seeds from the oil industry. So there are different things that we're looking at, but my big passion is legume water, and I would like to explain to you what the options are for the food industry and also for every common household. And before I get into that, as Katie said in the presentation, I'm originally from Parma. So I've always been studying food science and technology because my idea was I'm gonna get a job in Parma because we have a lot of food companies. And then I ended up traveling the world. I first started studying food chemistry, and then I moved to product development, so physical properties of food. And this brought me to OSU, where I did a PhD with Dr. Votivot. And this is an interesting part for this project because I've been working on saponin as a way to reduce cholesterol absorption in food. So we were developing functional foods by adding saponin extract. And this became very useful later on when I started working on Aquafaba because I realized that Aquafaba is actually a source of saponin. So I used that basic knowledge and applied it to a newer idea. And the industry experience was for a gluten-free company. And this is where I learned a lot about hydrocolloids. And that's where I came with the idea of transferring Aquafaba to a niche market, which was basically vegan products or egg-free products, to actually something bigger, which is food ingredients, because I could see the potential in using it as a hydrocolloid or an emulsifier. So in the first studies, I focused a lot on gluten-free just because that's what I know best. But hydrocolloids are used pretty much in every food product, from dairy to meat to bakery. And so there's a lot of applications for them. And because I like R&D, I focus on sensory analysis a lot. So when I work in Denmark, it was a project on making bread out of cassava, because it's very common in places like Brazil, more common than wheat. So it's a more sustainable solution for them. But obviously cassava doesn't have any gluten. So in that case, we work not with hydrocolloids, but with enzymes. And that's another experience that I treasure, because having learned about enzymes, I can now see how there is a potential for them in reducing food waste, because enzymes can be used to change the functionality of food, to improve their texture, but also to improve their bioavailability of nutrients. And after that, I finally came to New Zealand and I stopped traveling, at least for a few years. I'm teaching the beer class and principles of malting and brewing. But I'm also teaching food innovation at bachelor and master level. So I came here to teach, and obviously being new to town, I started traveling around a little bit. So for those of you who don't know, we are really, really far from everyone else. We are here, a tiny island in the middle of the ocean. And it was a big change from OSU, but there are actually a lot of positives. I'd like to share with you just a couple of pictures that I took recently. For instance, this one on the left is Lake Clearwater in the South Island. This one on the right is Lake Tekapo, which is very, very famous. And there are a lot of beautiful sceneries to be enjoyed. And specifically, I live in Christchurch, which is in the South Island. And here, when you say south, you say cold. So it's colder than the rest of the country, but it's still a very temperate weather. We rarely go below freezing. And we're lucky to be close to mountains and the ocean. So we get a lot of sceneries like these ones. And that's something that I enjoyed a lot. And I think it does help with innovation, because if you take a stroll on a place like this, it helps clearing your mind. But obviously the negative was when I came here, I didn't know anyone. So I started going to some events, to some meetings, to get to know people. And one of the first things I did was going to a dinner party where people were looking at alternative food products. And that's the first time that I heard about Aquafaba. So that was at the end of 2015. Aquafaba was already a thing for at least a year at the time. And basically, for those who still don't know, if there is someone who doesn't know, basically Aquafaba is a Latin term for bean water. And it refers only to the brine of chickpeas. So chickpeas are usually canned and cooked in the can. That brine was took by a chef who actually realized that he could whip it into a mousse and later even bake it into a meringue. And at that time, people knew this, but nobody knew the science. So I was very interested in figuring out what happened. And basically the key to this is that there are protein, but also phytochemicals, the saponins that I studied at OSU, they help with the foaming ability, but there are also fibers that help with the gelling property. So this is when I started to think and connect the dots and say, oh, maybe I can use it as a food ingredient, not just as an egg replacer. And we did some research and we mostly focus on a few nutrients. The proteins, we realized that most of the proteins are soluble, which helps with the foaming ability. At this stage, we do know the protein content, but we don't know much about what type of protein and peptides we have. And so, spoiler alert, this is the type of research that we will be doing in the future. We also know that there are saponins and they help stabilizing the foam. So just to give you an idea, this is a test that I did myself. I'll lower the volume a bit. And you can see the moment you start whipping this chickpea brine, you see a lot of foam. That is the protein incorporating air, but it's also the saponins that allow the air to stay there. So at the end of the process, you will see that that foam actually is extremely stable. And even after I stop whipping, it's still there, still perfect. So this explains the foaming ability, the soluble protein, the saponins. It doesn't explain how it bakes into a meringue. So we did some other studies. We look at the emulsifying properties and we look at different legumes because maybe it's not just chickpeas. So we try to have a representation of all the different legumes that industries produce. And we look at haricot beans or navy beans, but also lentils, split peas, and later on also soy. And we did realize that pretty much all of them have a very strong emulsifying ability. This picture is from a very basic test. We mixed equal volumes of legume water and oil and homogenized it with a homogenizer. And then we centrifuge it. So after centrifuging, this white layer that you see in the middle is the emulsion that is still stable, while the color part is the water that's separated. So you can see that a lot of different legumes have this emulsifying ability in their water. Another thing we noticed is that the oil layer that was expelled was really, really small. So this suggests us that this is a type of emulsifier that works well for water in oil emulsions, as opposed to the other way around. So that was already a very interesting information. So it could work very well in systems that are higher in fat. And we also show here that it's not just chickpea, but it's also other ingredients. And I believe that here it's a combination of different proteins, but also saponins that stabilize the emulsion. And then we look into the gelling ability. So we use the meringue just as an excuse to test whether it can gel or not. And this picture looks decent, pretty nice, but this is chickpea, and also pea gave similar result. When we tried to make meringue out of the water from haricot beans, we got pretty much a flat product. It was a complete failure. And we had other problems too with lentils in particular. So we managed later on to find a correlation between the gelling ability and the content of insoluble fiber. So there are different types of carbohydrates that leach into this cooking water. And those that are less soluble help with the gelling ability. And so this meringue experiment allowed us to understand why there is gelling ability and which nutrient gives that property. And that is valuable because this tells us that if you want to use these ingredients for foaming or emulsifying, we can use a lot of different sources. But if you want to use it for the gelling ability, we should focus only on a few, like chickpeas and peas, which also happens to be the best in terms of taste, because someone might wonder what the taste is like. They do come with the beany smell, but the whipping does help getting rid of some of the beany smell. And what's left is the bitterness, and that comes from the saponins. That's why only some of these ingredients work better than others. But this is something that is already well established now, so you might know some of these. And because of that, we actually already have applications for it. So originally, Aquafaba became famous as a vegan replacement to eggs. So you can find it in egg-free products. But it's now also becoming popular as a hydrocolloid. This picture here is from an experiment that we did a few years ago. This very sad bread loaf in here is a gluten-free bread without any type of additive. So we just made a basic recipe. And you can see in this picture on the right, this is from a microscope, from a surface electron microscope. And what we did is we took a picture of the bread crumb inside the pores. And inside the pore, you can see that the pore is very small, and you can see a lot of black patches. These are holes. And this is a very common problem with gluten-free products, in the sense that gluten-free products don't have the gluten starch matrix. So during cooking, during the baking, the water gets released and lost. And that's why they taste so dry. Then we replace the tap water in the recipe with the chickpea cooking water. And we got this beautiful picture here. So now the inside of the pore is much larger, which suggests more volume. And in fact, we did have a higher volume, but also it's nice and smooth without those holes. And this was likely because of the gelling ability, the fact that the insoluble fiber gelled and allowed to block the air inside the bread, thus making it juicier. And we're not the only ones who found out about this. In fact, if we look at it right now, in terms of industrial application, we already have a few companies selling Aquafaba as a commercial food ingredient. There is a big company in the US that just started less than a year ago to sell Aquafaba as a liquid. And I put a link here for those who are curious about which company this is. But there is also another company that is selling it in a powder form, because Aquafaba has roughly 90 to 95% water. So if we can dry it into a powder, obviously there will be more applications. because we don't need to worry about adding extra moisture to food. The challenge is which drying method to use and how that affects taste and nutrition. And as a result, we already have companies also using it in their product. This picture is a mayonnaise from the UK. And what they did is they took the egg out of the recipe and added aquafaba. To be fair, I also noticed that they added acacia gum and xanthan gum. So they did add some hydrocolloid to stabilize it, which suggests that the recipe is not optimized yet. And the reason is that, yes, there is foaming ability, but it's lower than in egg white. There is gelling ability, but it's different. So I wouldn't recommend aquafaba as a perfect replacement for eggs, but I would definitely recommend it for foaming, gelling, and emulsifying abilities. And the fact that there are companies already selling it like this is really encouraging to the movement. But this brings us to the interesting part, which is what can we do next? So because I like to work on legumes, and because here in New Zealand, but also at OSU, the dairy industry is very big, we decided to look at the soy and dairy industry in terms of processing. So things like yogurt, cheese, tofu, any type of food product made with these, with milk or with soy milk, and look at what's the environmental impact. So in this review article, we look at a few aspects. The first one is the environmental impact. And basically, when we process these milks into products, we do have production of wastewater. That could be from soaking and straining, or could be, for instance, whey production. And it really depends on the process, but we're talking about several liters of water that gets wasted. And that is a double problem. It's a problem for the companies because they need to treat this water before disposing of it in the environment. So obviously that brings costs. These numbers are just estimates, but obviously it's a cost for the company to treat these waters. But it's also a problem for the environment because these waters are rich in nutrients. So a couple of terms that you might be familiar with are the BOD and COD. So BOD is the biological oxygen demand. And basically that is a way to estimate how much it takes to clarify a product before disposing of it in the environment. And this focuses on microorganisms as a way to treat it. So usually wastewater is treated either with microorganisms or with filters and membranes. And you can see very high numbers, even more higher for the dairy industry. That's because dairy byproducts tend to be higher in nutrients than legume byproducts. But the point we are trying to make here is that we know there is a lot of nutrients in here. So it's causing cost to the companies, problems for the environment, and it's also a loss of nutrients. So we already know success stories like whey being used to make different type of products, but also to make protein powders for beverages. So I thought, why not doing the same thing for legumes? Because that's what I work on. So to give an example, if you think about tofu, in order to make tofu, we need to take the soybeans, soak them, and that generates soaking water, 2 parts of soaking water per part of soybean soaked. And then they have to be cooked and drained. And then again, there will be wastewater in there. And it's not just soy, but it could be chickpeas made into hummus, or it could be other things like sprouts. So mung beans are very common to make sprouts. And when we make sprouts, basically we are adding water to our beans, soaking them, and then letting them sit at warm temperature for a long time. This generates wastewater again. This picture on the right is a not so sophisticated system that we use for our experiment. Obviously in the industry it's different, but again, there is wastewater produced. So this gave me a general idea, and this is what I call Liluva. Basically the idea is, at this stage we're using brine water from chickpeas, but all the different industrial processes also involve soaking, boiling, straining, like in the case of tofu, sprouting for the sprouts. And it's not just chickpeas, but it's different type of beans, like haricot or mung beans, lentils, split peas, soy. All these different legumes, all these different processes generate wastewater. And this gave us the idea to look into all of this. And that's also where we came with the name of Liluva, just to come up with a general term that covers all these type of waters, instead of calling it wastewater, because obviously it didn't sound very exciting. So we just come up with a short name to describe it. And I believe that this will be the future, because Aquafaba is already a thing, but we know that the industry is also looking at other options. There are a lot of companies that work with peas, lentils, soy, and they have byproducts. So we did research of this. And the first thing we notice is that it's not just Aquafaba that has foaming ability, emulsifying or gelling. So we found this, as you can see in the pictures, for haricot, lentils, peas. So it's really just a matter of choosing the right legume source based on the flavor that you want to have, because chickpeas and lentils tend to give very earthy flavors. So I can see them more appropriate for savory foods, while things like haricot beans and split peas are more on the sweet end. So they're better for sweet applications. It really depends on where you want to use it. And also, because there is a big demand for cleaner labels or for simpler formulations, I think another application would be the replacement of hydrocolloids, because that will also allow the cost of the recipe to be dropped. Some of these hydrocolloids are very expensive. That's something I saw in the industry experience, having to source hydrocolloids from specific countries. Sometimes there is a shortage in the supply chain. So if we can replicate a similar functionality with whichever wastewater we have in our country, that could be a great advantage. So what I did here, for instance, I look at the gluten-free bread, and I used only one legume to keep things simple. And I look at soaking and cooking water. You can see from the picture straight that the volume has increased quite well. And it also looks like it's cooked a little bit better. We measured texture with a texture analyzer, and the results were amazing because the hardness was pretty much half. And that was very interesting, especially for soaking water, because the soaking water of chickpeas has pretty much almost nothing in there. Like the amount of solids is roughly 0.5%. It's very low. So you wouldn't expect much of an effect from such a tiny amount of solid, but we did see a huge drop in hardness. And then we thought about it and we realized, well, when we soak chickpeas, what happens is that we have sugars, soluble fibers, and also some soluble protein that leach into it. And all these soluble carbohydrates, and to some extent also saponins, are very good emulsifiers. And emulsifiers are known to soften food and to help incorporating more air. So that explains the bigger volume and the higher softness. For the cooking water, even though the numbers are similar, the taste was completely different. It felt a little bit harder. It definitely felt juicier at taste. So we believe that here it's not about emulsifying activity, but it's more about the gelling ability of the insoluble fiber that allows the air to be inside. So two different mechanisms, but similar results. One thing I'd like to say though is when we use the cooking water, we did notice an increase in saltiness. And that's because there are a lot of minerals in this ingredient. And in particular, chickpea is very rich in sodium. So it does bring the salty taste up. A simple solution to that is to reduce the salt content in the bread recipe. So hydrocolloid is one idea. And here again, you can see from the picture why I'm talking about different mechanisms. With the cooking water, we have a nice smooth pores, as we saw earlier. With the soaking water, the pore is bigger, yes, so there is more air, but we still have a few holes. The only difference is that these holes are not as deep as for the control. So this is telling us that with the soaking water, we don't have enough gelling. And in fact, when we tasted this bread, it was softer, but it still tasted dry. It was still a bit of a cardboard type of taste. So this helps us understanding the different mechanisms. So I talk a lot about texture because that's what I like to work on, but I believe that we should also look into nutrition and in particular bioactives. So the key thing about these ingredients is that the amount of solid in there is relatively low, but they're very powerful. So all this water contains saponins and phenolics, and saponins are present in very high amounts. We are talking about anywhere between 10 to 15 milligrams per 100 gram, which is quite a lot. And there are different types of saponins, and apart from affecting texture, they also reduce the absorption of glucose and cholesterol. And so this brings the idea of using some of this wastewater, for instance, to look at the effect on cholesterol and glucose metabolism. And this is what one of my PhD students will be working on, looking at whether there is actually a positive impact of using this bioactive. And the cool thing about this is also that there are a lot of companies interested in extracting saponins and adding them to products. If we use this water, we don't have to go through any extraction because the soaking and the boiling, it's pretty much already an extraction on its own. So that will make it easier and cheaper. Another very interesting thing we discovered, there are a lot of minerals in there. We have a lot of iron, particularly from soy, but we also have potassium. That's the main mineral we found. And to some extent, we have things like calcium and zinc. So all these things are very interesting in terms of nutrition. They're also interesting in terms of taste. That's when I mentioned that the chickpea ingredients tend to be saltier in flavor. But I think that could be an excellent opportunity, for instance, for isolating nutrient. And this will go in support of the supplement industry. And then the other thing I wanted to mention is the peptides. So up until now, we know the total protein content. We don't know exactly which proteins are in there, but we have some very good guess for what will cause the foaming ability and the nutritional properties. And on top of that, we have enzymes. And so I'm going to talk about that in the next couple of slides, because I think that considering the big demand now for plant protein, this could actually be a very interesting solution, because plant proteins, again, are hard to extract. Some are soluble, some are not. In these ingredients, because they're water-based, the proteins are mostly water-soluble, and they're easily accessible. And so this is the idea that we have now, to still work on texture, but also look at the nutritional benefits of these ingredients. So it's no longer about egg replacement or hydrocolloid replacement, but it's just about taking advantage of these ingredients. So instead of considering it as a waste, just actually use it and take it to our advantage. And then the last thing we notice is shelf life. So another approach to reduce food waste is actually to guarantee enough shelf life to food so it can last long enough. And we look at that in two ways, in terms of sensory and safety, micro. In terms of sensory, what we did is we had an experiment on crackers. So we made a control cracker with regular water and the soy one with soy water. We measure hardness. At day 0, they were kind of similar, slightly harder the soy one, but similar. After storage, the control cracker became harder, and that's obviously because of staling. So the starch retrogradating, becoming harder, and also moisture loss. So all of this is normal. But the staggering results was for the soy formula, because not only the hardness didn't increase, but it actually decreased. And we replicated this experiment at least 6-7 times because I couldn't believe this number. I thought there was a mistake. And just to explain how we stored it, we put the crackers in plastic bags and stored them at room temperature. So it was not a sealed packaging. It was not modified atmosphere, just a regular plastic bag. And so we look into this, and then we realized that actually the moisture content in the soy cracker increased over time, which I have to say for the crackers is bad, because we want crackers to be crunchy, not to be soft and soggy. But still, it led us to an interesting discovery that actually we can use this soy water to increase water absorption and keep bakery products soft. It is strange that it took so long for the soy ingredient to absorb water, but the reason is probably in the fact that soy proteins are very different from the other legume protein. Soy protein like to absorb water, but they take time to hydrate. So it is likely that it took at least a day for the soy protein in the soaking water to completely hydrate and start absorbing moisture from the atmosphere. So for the crackers, that was bad. But if we think about bread, that could be an excellent result. And other things to consider also are the saponin and the protein content. But to be fair, saponin and protein content in this ingredient are quite low, while they're higher in other ingredients, and we didn't see the same effect. So I think it really has to do with the type of protein found in soy, which is why now we're so interested in what type of protein we're dealing with. And then the last thing about food being stable is obviously whether it's safe or not. So very simple experiment. We made some whole grain bread, no longer gluten-free, just regular bread. And we replaced, again, the tap water with some of this Liluva. And the reason we chose beans and peas is because these are the ones that have the least impact on sensory. And then what we did, we just let it sit at room temperature on a desk and wait for it to go moldy. And so for the control and the bean bread, it took 6 days before we could see the mold. For the split pea, it took actually 9 days to see a little bit of mold that also happened to have a different color and a different shape. So most likely it's a different type of microorganism, but it's definitely a much longer time before it went moldy. And that is very interesting. And we look at all sorts of explanations. Saponin are known to be antimicrobial, but we couldn't correlate that result to this one. So probably it's not the saponins, because typically they inhibit bacteria, not really molds. We look at a lot of other things, phenolics, but they're not that high. And then we came across these interesting papers talking specifically about peas and lentils as a source of a particular peptide called defensin. And this peptide is known to be specifically active against mold, while it does not inhibit other bacteria. So this becomes very handy for bakery products, because it's telling us that if we really have that peptide in here, and we're going to find that out soon, if we do have it, then it allows to inhibit mold without affecting yeast for bread making. So that didn't give us any problem, and without affecting other bacteria. For instance, we tried also some of this water, which are higher in saponin content. They went moldy at the same time as the control. And on top of that, the bread was not that good because having too much saponin can affect the yeast and the fermentation of the bread. So having this particular peptide is very cool because we can slow down the mold without affecting the sensory quality of bread. And I thought that this was a very interesting result, and it's part of the reason why we're really looking into it right now. So I know this is a lot of different things, but it's just to show you the potential, not just egg replacement, but also hydrocolloids, shelf life in terms of sensory and in terms of molds. The last thing I want to mention is the enzymes. Speaking of peptides, if we have protein, there is a chance that we have enzymes. Maybe not in the cooking water, because having been boiling for that long, probably those enzymes are inactivated. But when we look at soaking and sprouting, these are wastewater that have been treated at room temperature. We know there is protein, so there could be some enzymes. So what we did here, we used a simple colorimetric assay. Basically, in this plate, we have 2 different substrates. The red one is specific for protease enzyme, and it was a casein substrate, while the blue one is specific for cellulase enzymes. It was based on cellulose. And basically all we did is taking our samples, putting them on this plate, and then incubating. We followed the procedure suggested by the manufacturer of this kit. And after incubation, we measured the absorbance with a spectrophotometer. And we did find the presence of enzymes. You can see that also by the different colors. And the reason this color is darker than the higher one is because these are different samples. And we look at the few ingredients. And what is interesting is that for some wastewater, we found a high amount of protease, while for other ones, we found mostly cellulase and potentially either amylase. So it's a combination of different enzymes. Obviously, we found more than one at the same time. But the interesting thing is that each wastewater had a different predominant enzyme. So based on the need, whether we want protease, cellulase, or amylase, we can select the right type of wastewater. and then use it as a source of enzymes. And this is showing us analytically that the enzymes are there. The reason we went all the way there is because before that, we ran some practical tests. And we made different food products where we added a source of protein, like whey, or a source of fiber. For instance, we used spent grains from beer making, the spent barley, which has a lot of cellulose. We made some food products with them, things like cookies or bliss balls, different simple products, and then we treated them with our Liluva water. And we did notice a change. Basically, the key thing we noticed is that, for instance, when we had a high fiber product and we added an ingredient rich in cellulase, we measured the texture and we noticed an increase in adhesiveness. And adhesiveness is a property that is measured with the instrument that give us an idea about juiciness. So a food product with high adhesiveness usually tends to be juicier. We did that for cookies. We did it for these bliss balls, which is basically a mixture of nuts and dates. And same thing when we treated the high protein source with protease. So that really is showing that the enzyme is working. And I think this has huge potentials for two reasons. One is the enzyme industry, but they may or may not be interested because maybe it's not a problem for them to find enzymes. But the biggest application, the second one to me, is the utilization of byproducts. So for instance, we look at the spent grains from beer making. There are companies that are making granola bars and other foods with spent grains. The problem is that there is a lot of insoluble fiber. So it's not very nice to eat, obviously, and it's hard to digest too. So if we treat it with the cellulase enzyme, then we can break down some of the fiber, which means that there will be more nutrition available, but also the texture will improve because it will become juicier. And so I think that there is a lot of potential here to combine this particular ingredient with ingredients that are high in insoluble fiber or protein, particularly when we look into plant-based food. So whether it's high cellulose or high insoluble fiber, and this is a very common problem, if we treat it with this type of enzymes, we're basically combining two different byproducts, and we can get very good results in terms of nutrition and taste. So I think this is the ultimate future application, and on top, of course, of all the other ones. So I'm very excited about all these applications, and just to put them in context, at this stage, nowadays, we already have Aquafaba available in the market as an egg replacer or as a texturizer, like hydrocolloid replacement. What I foresee for the future is that soon we will start to see also other ingredients based on different legumes, like beans, soy, lentils, peas, mung beans, and based on different processes, because Aquafaba involves cooking. So it's mostly for house consumption or for companies that cook their own chickpeas to make Aquafaba. But our idea is to look at what food companies produce as a waste. So usually the waste comes from soaking, sprouting, and only to a minor extent comes from cooking. So we are looking at what the industry produces as a waste, so we can minimize the waste, and then we can repurpose it. And it's not just egg replacer or texturizers, but it's also improving the shelf life in terms of softer texture and also delayed of molding and spoilage. Another thing I haven't mentioned here, but I will do now, is the source of prebiotics. These ingredients contain complex soluble carbohydrates, which are excellent prebiotics. And we did run this test. We noticed very high prebiotic activity in most, but not all of these ingredients. And the reason is that we also have to account for anti-nutrients and for different conditions. But we definitely saw a huge potential for the production of prebiotics. Now the industry is switching, at least here, I don't know in the US, it's switching from probiotic-rich food to prebiotic-rich. So now in the supermarket, there are a lot of products with the label high in prebiotic or source of prebiotic. So I think this could be an example. And we can also look into other applications. For instance, there is a university in Singapore that has work with tofu whey, and they fermented it to make food product. So that's another idea. I can see why they came up with this idea. We have prebiotics, we have nutrients, proteins, so it could work. And then the last thing is a source of bioactives, which is not just phenolics and saponins, but it's also specific protein and specific minerals. The last thing is using it to make edible packaging. We know that these ingredients contain a lot of protein and a lot of fiber, and they're mostly soluble. So if processed correctly, there is a potential to make it into packaging. And I think this will be interesting because that way we don't have to use food to make packaging, but we use food waste. So we are actually not taking away food from the food chain, but we're just repurposing a byproduct. And the challenge will be figuring out how we need to process it, whether it's with enzymes or with other additives. And the edible packaging is the dream, the vision. If that's not possible, biodegradable packaging that is not edible, that could be a step one to make things easier. And so you can see how there are so many numerous applications. And these are the ideas that we came up with and the results that we got. Obviously, there are other possibilities that we may have not think of. And for most of these, we have a good knowledge, but now we still need to get into the details. So in our research, now we're focusing a lot of the health benefits of this product. We and other researchers in the world have spent now about 4 years working on this ingredient as a texturizer. There are quite a lot of publications out there, and people in Europe, North America, Oceania, Asia, pretty much everywhere, South America as well. But I think the nutritional part is very important. We don't know as much about nutrition, so we would like to focus there. And so the ultimate message to me is that if we can repurpose these ingredients, first of all, we're cleaning the environment because we're producing less waste. And second, we are saving more water, which is another important aspect. So we don't necessarily need to dry it to a powder, but the fact itself that we are reusing this water as opposed to wasted, that's a big issue, because every now and then most countries have to deal with the issue of not having enough water. And by cleaner food, what I mean is we can use some of these ingredients to improve texture and shelf life without having to use additives, which, yes, will give a clean label if you want to call it that way, but it will also lower the cost of production. And I think the biggest challenge here is how to go from an experiment to industrial application. The companies that are selling Aquafaba right now, I'm not sure how they do it, whether they source the byproduct from, let's say, a tofu manufacturer, or whether they produce it themselves. Obviously, that would be the challenge, the industrial scale up. But there are ways around that, either with startups or with industry collaboration. And that is something that we're looking into right now. So this is what I wanted to present to you today to introduce the topic. And before I close, I would like to thank my huge research group. So as I said earlier, I just started supervising PhD students, but most of my work in the past has been done mostly with bachelor and top master students. And they all do the equivalent of what would be considered an honors project at OSU. So you can see how many people I've been working with, and this is not all of them, it's just a small representation. But I would like to thank them for their contribution to our research projects, to all the papers. And I'd like to suggest a few references that I used. There is an interesting book chapter about Aquafaba, and then from a research group in Canada, and then also our book that we published this year in the middle of the pandemic, so not the best time to come up with a new publication. But this is the book where we talk about all the different waters: soaking, cooking, sprouting, bioactives, packaging, all the different applications. And I also like to keep in touch with those who are interested in the topics. So I have a series of social media that I use, and also my email address is there. And at this stage, I would just like to open to questions. If you have any questions for me, and I'll be glad to start the conversation about Aquafaba and Liluva. Thank you.
Thank you. So if anyone wants to just unmute themselves and ask a question, or you can type in the chat too if you're comfortable. Any questions?
Sure, I'll ask a question. Thank you for that presentation. And I love your Instagram handle. It's like Dr. Legume Water. It's very creative. So my question is about these enzymes that you talked about in legume water. Are you concerned about them breaking down the inherent proteins in Aquafaba or Liluva and leading to the formation of some bitter peptides and off flavors?
Did we lose him?
Yes, I think we did.
He not only quit the screen, I think he quit the presentation.
Yeah,
Maybe his internet went out. We'll give him a second.
yeah.
He'll sign again.
Mhm.
That's a really good question. The anti-nutrients that, and I want to ask him about the saponins.
So Dr. Menes, are you working with Aquafaba or like dairy wastewater?
I was just part of, trying to help. I was part of his grant. So we're trying to do since we're doing now a lot of the biological activity components, that's probably what I work with. And just like Alba is doing whey with fish waste, I like the idea of combining, whey with Aquafaba to do different textural ingredients.
Mhm.
Both things could work, even when we have proteolytic components. The ones that I'm interested, like in the saponins, they're really interesting because in your stomach, they make a complex with cholesterol-like components. So these will make you secrete the bile salts, therefore lowering your blood cholesterol. Because normally bile salts get reabsorbed, and it has nothing to do with your dietary cholesterol. People that have high cholesterol is because they synthesize a lot of it. And what you want to do is to have diets that make them degrade or excrete the cholesterol we make, which is always in the form of bile salts, bile acids. And the saponins are really good to do that. We do have a patent of removing cholesterol from foods using saponins. So if this is the source of saponins that is not too astringent, because that's the big problem with saponins, then this would be a really fantastic thing. And the other thing, I don't know if you saw, I hope that you did, is when he talks about making something soft, I think it's super interesting, the emollient properties that this has. Oh, he's back.
Oh, sorry, my computer crashed.
Oh, well, that's because you're there standing on your head in the southern hemisphere. Christ.
Upside down. Sorry about that. I don't know what happened. But...
So Aishwarya had a really good question. So I don't know if you heard it. But why don't you?
Oh, no. Could you please ask again?
Yes, sure. I was wondering about these enzymes or proteases that you talked about in Aquafaba and Liluva. So are they able to break down the proteins that are inherently present in Liluva and lead to the formation of bitter peptides? Because that could cause some flavor degradation and stability issues with the wastewater itself.
That is a very good point. We haven't looked into that. Right now, we only tested it on external protein. So it was whey protein that we tried and a few others from seeds. In terms of shelf life, we haven't noticed any change in the product in terms of taste. And we did a few things. We did freezing and thawing cycles. We saw the solutions being very stable. We also tried living in the fridge for a week or more. No problem. We have not looked specifically into that. although shelf life is something that I want to look into now. So one of the first things we are going to do now is to look at the stability of this product. So we're going to try that. Yeah, we're thinking about generically about the stability of the product, but it could be a good idea to look at protein, whether they change over time. They could.
Okay, thank you.
I'm giving a chance to the students to ask because I have many questions, as you may imagine.
Yeah. If they have any question, even the most generic one, because I'm sure there's a lot that has been missed in this presentation.
I'm just wondering about like the scale of how much of this water is produced, and like would it really be feasible to transport it since it's so heavy? Or would you like, are you recommending that companies are doing this on their premises? Like what kind of?
Yeah, so I think for the short term, the best approach would be companies actually doing it pretty much themselves. So it could be a company that makes a particular product like hummus, etc., and then also produce their own water and sell it. But at the end, it will have to be concentrated somehow. So we are looking into options of whether to dry it all the way to powder or whether to just concentrate it, because drying it could be a big cost, because we're talking about something that from 5% solids to maybe 0.5%. So drying it could actually become so expensive that it's no longer sustainable. So what we are actually going to do in the next few months is to look at different ways to concentrate it and look not just about the quality of the ingredient, but also the industrial feasibility. So things like cost and energy input. And so hopefully in about 3 or 4 months from now, we'll be able to give you a very specific answer on that. But I think that it would be a compromise, like trying to reduce the amount of water so it's easier to carry it around.
Yeah, it's interesting. If it's already on the market, it's definitely feasible.
Yeah,
yeah.
I was a bit surprised by the one that they're selling as a powder, sorry, as a liquid, because the powder one, depending on which technique they use to concentrate it, could be feasible because it's a byproduct. So even if it takes some energy to dry it, it's fine. The one they're selling it as a liquid, my guess is that it could be maybe just a first attempt to get it on the market and see if consumers like it. And then maybe in the long run, they might change something. The good thing is that we don't need to use a lot. So there are some ingredients that are sold. I remember when I work in a dairy company, we work with liquid eggs, for instance, stuff like that. So maybe it's a similar approach. But yeah, now we can see that it is feasible.
Mm-hmm. Thanks.
I do have a question, a burning question, Luca, just because of the surface activity that you're observing. Are there any other components like phospholipids or waxes or things that are normally found in in the surface of beans that are helping in that surface activity? So that's one. And the second question is, what tests do you have with the saponins to theorize that they can remove cholesterol?
Yeah, so regarding the surface activity, we do know that there is no trace of lipids in this water. There could be some fatty acid or some lecithin. We haven't looked into it, and just honestly is because with the resources I had with the bachelor project, it was mostly looking at proximate composition, basic stuff. Now we're looking into using HPLC. That's a good suggestion, because if we just correlate emulsifying stability and activity to saponins, it is not a linear correlation. There is something else. And the picture I showed was one type of test, but we also use another test based on spectrophotometric absorbance of the emulsion, and that gave different results. There were big differences among samples, and we have to understand it could be the presence of some lecithin maybe. And in terms of cholesterol, so we did a few studies. We studied with Keiko cells. We saw that the absorption of saponin is quite low, so they don't really get into our bloodstream because it's only about 1% of the saponin that we eat that actually gets absorbed. But we did notice a lower micellization. So we did in vitro models with Dr. Failla, and then we look at micelles, how much cholesterol goes in the micelles. And we did see a reduction based on the saponin type. So it could be that the saponin replaced cholesterol. That's one option. And the other option is that they might increase the production of bile acids. We did not test that in our experiments, but it's been proved before. So it's competition for the micelles, increased bile acid. The third option could be simply more gastric emptying. So they just create a bulk. And that is what we want to figure out now, which one of these mechanisms is taking place.
Yeah. Exactly. I think this is, well, I just want to thank you. It was a very, very interesting and original approach to research. So
Oh, thank you.
We have to keep that connection with Ohio State. Just remember that.
Yeah, so I've been talking, for instance, to Ken Riddle because he works a lot on phytochemicals. So we have been working on saponins before. And now I only know the total saponin content. I don't know the profile. So that could be the first thing to look at. But then also definitely I'm interested in collaborating, and I'm collecting some funds. So the way we can do something a little bit more interesting with more research fund. We did secure some for one student. I'm going to have another one starting hopefully in March. And so with this, obviously we can do more.
We need to talk a little bit because, you know, Harjendra Singh in the REDET Center is very interested in this kind of thing. So I think that we should put you in contact, and they're there in New Zealand.
So what is the name?
In Palmerston North.
Yeah.
Harjendra Singh. He's in the REDET Center, Massey University.
Oh, okay. I've been talking to someone at Massey. I don't know this person, but we'll contact them as well.
Yeah, well, we'll talk about it because Harjendra is the director and he's a good friend.
Oh, that's very good. Because in terms of resources here, we mostly look at composition. We did get some advanced equipment like a mastercizer, but it's not the same as Massey. They're more into technology. And with OSU, I'm looking at collaborating on nutrition and protein and phytochemicals.
Let me know if we can help.
Yeah, definitely.
All right.
Awesome. Well, if there are no other questions, thank you so much for being here with us. And everyone have a great rest of your night.
Thank you very much, Luca. Bye.
Thank you. It was a pleasure meeting you. Bye-bye.
Bye.
Bye.